Hello, everybody, and welcome to the PhenomicsWebinars. My name is Philip von Gillhausen from IPPN, and today's topic is on digital phenotyping of individual plants and plant communities in the field. For this, I want to welcome Professor Robert Juncker from the University of Marburg and the University of Salzburg. Here with us today. Welcome, Robert.
Thank you, Philip, for the invitation and for the chance to present my work here, or our work, I have to say.
It is our pleasure. Well, let me shortly introduce you very, very briefly, because actually we know for quite some time. I think our first contact was around 3 years ago. When you decided picking up digital plant phenotyping for ecological studies, which is still a path that is not walked very often so far. Anyways, I'm always very happy and very interested if digital plant phenotyping methods get to apply to somewhere out of bounds, out of the traditional, so to say, applications like plant breeding, for example, or genetics. So, Robert, maybe you want to explain us a little bit on what brought you first to applying plant phenotyping methods in ecology, but maybe even before that, please explain us a little bit about your work, about your position and yourself.
Okay. Yeah, thank you, Philip, again for the invitation, for the introduction. And I'm very happy to be here and also to be able to talk to you live, not in person in the same room, but live, because as you may have noticed when watching the recorded talk, that is something, the tension is missing. So yeah, maybe I couldn't transport my excitement for our work well enough, and maybe today I have the chance to do that. So first, regarding the confusion about Marburg and Salzburg. So I was working for a long time in Salzburg from 2013 to 19. And in 2019, I started a new position in Marburg, where I'm now a professor for evolutionary ecology of plants. But I'm still affiliated with Salzburg, so I'm still a group leader there. So both is true. And so as you saw no, you didn't saw it in the talk, but I have some pictures today. Maybe I have the chance to show it. We will apply the machine, the plant phenotyping machine, also in the Alps this summer, hopefully. And so that will be the next adventure with bringing it really to the field. So why I'm interested in plant phenotyping, ecology or community ecology emerged from species-centered approaches. So people measured how individual plant species respond to certain environmental gradients, how they interact with other species, and how they perform in certain environments. And that is and that led to very strong insights in plant ecology. But the problem is it wasn't really generalizable. So we knew this one single plant species is responding to drought in that way, or to climate change in that way. And so it is very hard to make predictions about the future, about other communities, about communities where the plant species is part of, but maybe there are other plant species involved in the same community, and they may respond differently. So we don't know that. And therefore, the trait-based approach emerged in the last, let's say, 10, 15 years, where people started to track the functional responses of plants and plant communities to environmental gradients. So let's say, instead of looking at a plant that is maybe performing less well under climate change condition, we are looking at how plant traits, independent on the plant species itself. Of course, we look at the plant species still, so we do not ignore taxonomy, but we look at the traits and see how trait composition, trait diversity, and the mean community phenotype is changing in an environment. And yeah.
Sorry to interrupt. So with mean community phenotype, you mean the functional identity of the whole community without directly referring to species names at least, but rather taking it from the, as you say, functional side by, for example, representing certain ranges or spectra of leaf area or certain leaf traits or reproductive traits and so on and so on.
So methodologically, you're right. So first what we do is we look at a community, we look at the plant species that are available at that community, and then we measure representatives of these plant species for, let's say, leaf area or specific leaf weight or specific leaf area, plant height, and so forth. And then we take these species-specific data to calculate, for example, a community-weighted mean. That means that is the mean leaf area for all the plant species weighted by the abundance per plant species. And then we have one measure for the average leaf area of the plants in this community. And then we can also calculate functional diversity, which is the variability within a community. And that is really interesting because this functional diversity has been shown to be strongly correlated to many ecosystem processes. And so we assume that the more diverse a community is, the more resilient it is, the more productivity it can have. And so this functional diversity became a very important part of community ecology or trait-based community ecology, where we look at these patterns and how this diversity of traits is dispersed in environments along gradients and so forth. And so I think that is the advantage, as I said, of these trait-based approaches, because now we can look where in the world, where in the Alps, where whatever, can we find similar diversities? Does this diversity or these community-weighted means, are they reduced by certain drivers, or are they increased? And what does it mean for the environment? And then we can make general, or we can present general findings. And we can predict future events. And that is really, why we are so interested in plant traits.
Yes. I know from the information I got about you that you're not only working on an observational scale, as you described just now, so really going into the ecosystem and observing, measuring, identifying patterns which are maybe similar or generalizable, but you're also working experimentally.
Mm-hmm.
Can you tell us a little bit about what kind of experiments are you doing and what the purpose of these experiments is?
Mm-hmm. So maybe I have to talk about two parts. So we did experiments where we did this manual phenotyping. So with a caliper rule or a scale or something, where we just measured the plants in a traditional way. And here we were interested in even more generalizable results. So because here we can specifically change individual factors like the temperature, the moisture, and so forth. And then we can track how the plant species react to that or respond to these environmental factors. And now, as you also saw in the talk, we transplanted whole plant communities to a common garden in Marburg, here in the botanical garden. And that is, of course, also an experiment, although we don't bring it to the lab, but we bring these grass sorts to a common place where they have exactly the same environmental conditions. But what we do is we split each of these plant sorts soil sorts or sorts in 4 parts. And then we treat each of these parts with 4 different land use treatments. So we fertilize them in different ways. We mow them in different frequencies. And so then we have a full factorial design of mowing and fertilizing. And now we can track again the response of the traits and also by the species in these plots. And due to this splitting of the original sorts in 4 experimental parts, and because we repeated this with 39 plots, so we have 39 times 4 plots, and I guess the species responses, they will be different depending on where they come from. Let's say we have a species that is used to low nitrogen levels. If you fertilize them, the species will disappear sooner or later. But this is specific to this species. And when we look at the traits again, we can then learn if all the species respond, or if all the communities and their traits respond in a similar way to the same treatments. We can say, okay, this is what we expect, and maybe species that have rather these traits, so towards this this shift of the trait space, they may be favored by this land use treatment, and that is one part of the experiment. And
also gives implication to land use management.
Yeah, sure. Yeah,
ecosystems.
Yeah, so. Right. So how can we improve or increase functional diversity? How can we increase species diversity as such, because that is also an important component, and we should make sure that we don't lose any more biodiversity. And these are then things we can test and report like that. Mm-hmm. We also did a lot of experiments on individual plant species. So here were interesting, or our interest was in regard how, let's say, interactions of plants and animals changes traits. Here we looked at the morphology, but also at the scent emission, which is not really covered by the digital plant phenotyping yet, but maybe. So that is also interesting, how the phenotype of plants is changed by interactions and how this is then affecting the ecosystem as such. So this is the other line of research we are following. And I showed you this strawberry project. Oh, yeah. That was in the talk, that was not really like the most innovative project. Everybody knows that drought and wind is affecting plant phenotype, but that also served for us as a project to test the potential of this plant eye. Yeah. So we needed to have a very clear experiment where we had clear expectations on the outcome to see how this plant eye compares to manual measurements. And in fact, the correlations are very good. So when we go there with the traditional methods or with the plant eye, we can see that we have very strong correlations of biomass, of leaf area, of things like that.
It's also a strong arm in the application of digital plant phenotyping methods, especially image-based systems that are used for the quantification of experimental treatments, yeah, and the reaction, of course, mostly on the reaction of the plants towards these treatments, yeah, and there, of course, you can imagine big agrochemical companies, like BASF and Bayer are applying these systems just to quantify in how far treatment X has an effect on species X and so on. So, of course, with the independence of these automated systems from the human bias or the observer bias, yeah. These systems are very well suited to objectively assess an extent of reaction of a certain treatment. And this makes this methods, of course, also very useful in other lines of research, as you say. Yeah, I think, Robert, we more or less recapped now in dialogue the contents, or at least some parts of the contents of your talk, to fully give you the opportunity to repeat some key points of your talk. Is there anything you would like to mention before we close the recap round?
Maybe I'd like to do an addition to what you said on the advantages of this digital plan phenotyping. I think this human bias is really reduced, but I think there are other advantages that are stronger than that. For example, so we did the strawberry project. We did this comparison in the common garden where we compared the results of the plant eye and the manual or traditional measurements. Mm-hmm. And all of the students that were involved in this research said in the end, and we just today heard 2 talks in a student seminar where they concluded in the end, okay, in the next project, I would definitely go for the digital plan phenotyping because it is much faster. We get much more traits in a shorter period of time. So one scan lasts for 30 seconds. So that is really quick. And so when one of the students did these measurements with a traditional way in the common garden, she was working for 4 months, every day, 8 hours, measuring, weighing the leaves and getting the leaves and all like that. And the scanning altogether took maybe, so one scan for all plots takes 2 days and then we're done. And that was with the old system before we rebuilt it. So now we have an even better suited system to have it really in the field. And so now I expect to be even faster. And so we could do 6 scan runs in a year compared to 1 traditional measurements by 1 student. So that is an advantage.
Yeah, it's certainly an advantage. And I remember very well from my time as a researcher myself, I don't know if you knew that I have been working in community ecology.
Yeah, you told me at that time.
Yeah. And I remember the hours and hours of species sorting, identifying, measuring or weighing different traits. It was so cumbersome and it was so error prone because after several days and hours in a day looking at plants, of course, you somehow only see green at a certain point. So I think, yeah, with the objective eye of a camera or of a sensor that does that in a portion of the time, I think a lot of value is gained. Yeah, not only on a qualitative dimension, but also, yeah, just plain work hour related, and this cascades down to euros per hour spent, and yeah, that is just tremendous. The improvement or the potential that this technology or this yeah way of applying the technology holds for within ecology, especially. Yes.
Maybe there's one restriction I'd like to mention, because I am an ecologist, so we need to value species, of course. One problem is, at least at the moment, I guess we can go there with artificial intelligence or machine learning, that we identify species or at least identify plant individual within these scans. At the moment, we are using values that integrate all the plants in this community, so in the whole scan area. And so if we are interested in community-wide responses and community-wide traits and characteristics, the scan, I guess, or I would say, is by far the better option compared to the traditional measurements. if we want to learn about species identity, species traits, which then could go to a database, of course, at the moment, this is not a replacement. So depending on the question, I think both approaches are still required in order to get a full overview about what's going on. But I think still regarding important ecological processes and properties of communities, the plant eye even provides more information than a traditional measurement. For example, biomass, which by the way is correlated very well with a weighted or with a weight biomass. That is something we cannot measure in the field, at least not in the Alps, in a national park. We are not allowed to mow the whole thing to weigh it.
Yes,
and so we can non-invasively get a totally new property without destroying anything. We just scan it, and then we can estimate the productivity of a given community, and we can even track that over time. And that is something we can't do manually,
yes,
or with traditional methods.
And it's
so you know, both methods have their pros and cons. And I wouldn't say we just go for digital phenotyping, but I'd say we need to include it in order to get more information about the communities.
Yes, so to say, to annotate the phenotyping data set with knowledge on species you have, that just the human eye can connect the information they see, and the species that displays this information can merge together then.
Yeah.
So, yeah, you said in the course of your recent work, you almost engaged in systems design with your new phenotyping tool you had, or you have, actually to adapt the system to be suitable, to bring it out to outdoors into rough environments. So you partnered up with the company providing the sensor that you are using currently, which is the Phenospex plant eye. And but with the off-the-shelf solution they provided in the beginning, you were not fully content. Can you tell us a little bit about the process from buying an off-the-shelf system to refining it, to adapting it, and then also to succeeding in applying it in the field, in that kind of rough environment in the Alps?
Yeah. Can you allow me to share my screen, because I have some pictures?
Absolutely. In the bottom, when you move the mouse trigger, you will find in the icons below.
Ja, Bildschirm freigeben.
Yeah, you simply start doing this, and it should override all.
Oh, it says the host. So you deactivated that I can share my screen.
Ah, sorry. Now it should be possible.
Now I think you see my presentation, right?
Correct. Very well.
Okay. So, yeah, that is a good question, Philip. So I never had the feeling that we bought something off the shelf. So I always had the feeling that we buy something that, of course, this is ready. So we bought it as it was. But the nice thing about Phenospex was the interaction with, so it started with you at that time, and then later mainly Alex took over, and that was a very pleasant experience, I would say, because yeah, we had so many questions, we had so many specific yeah, demands on the device that we really. So we knew very soon that the off-the-shelf solution cannot provide what we were hoping for. And so we had a lot of interactions, and that was very helpful and friendly and productive. And so I can explain you how it went. So this is what we bought. So that is provided by Phenospex. So that is not my slide. And I think this is what they imagined when they designed this thing. So they wanted to have a device that is transportable. So they fitted it in a suitcase. And in this one suitcase, there is everything included, and you can take it to the plane or in a car. And then within 10 minutes or so, you have set it up and you can use it. So that is what we see here. And I think for this environment, what we see here, that works perfectly well. So if you go to a greenhouse and if you bring the plants to the machine, that's perfect. So it's standing there, you don't move it, it's all good. But then we wanted to bring it in the field, and of course, in these communities, we don't have potted plants. We have like communities growing on the soil or from the ground. And so we can't place them on this tray here that you see here. And so we had to remove this. And this is, of course, very stabilizing for the whole system. And then it began to be very shaky. And over the time, it was making weird noises here at the transporter thing. I don't know the English term for it. You know, there is...
The actuator.
Yeah, so that one was doing crazy noises over time. And I'm saying this without blaming Phenospex, because they never intended us to do that. So we took their machine and, you know, took it to a place where they never thought about. So that was not their fault. It was our fault because we were too ambitious maybe at the beginning. And so we brought it there. And then after the summer, we thought again, okay, the results are cool and really great. We can do a lot with them. We can learn from these data. But how can we improve that? Because the common garden was in a semi-natural environment, I would say. So it was still close to the university. We had more or less even surface where we could place the whole setup. And this year, as I said, we want to go to the Alps. So my background here, we want to bring it there and measure it there. And you can see the surface is not even everywhere. We have to walk there for an hour to reach the sites. And then it needs to run the whole day without shaking, and it needs to be fast. And so we started to think about how we can improve this. And first I want to highlight Gabor Ferenczi and Michael Litzov. So Gabor is a technician in my group, and Michael is in the workshop at the University of Marburg. And they are ingenious engineers, and they helped a lot in improving it. And you see when you compare this, so this is the original frame. Now we totally, rebuild it. Maybe I go directly to this figure. So now we have this aluminium frame. This is the biggest advantage. So this piece here, that is stabilizing the whole thing. So that is new. Then we have this tripod, which is which can, you know, is good for rough surfaces. Then we got a new platina in German. What is it? So the electric stuff.
Yeah. Controller. Yeah.
The controller. So they gave us a new one. It is included in here. But otherwise, the parts are the same. Of course, the planter is the same. This, the word you said. This,
the actuator is the same. Yeah.
It's the same one. So that is now, it is very stable. It is easy to transport. And what we did is we had these barcode holders here.
Yes.
Because last year we had a lot of trouble in correctly placing them, and the planter is very sensitive to the correct position.
That's true.
And so it took us sometimes 10 minutes per scan to find the right position. And now Gabor built these holders here, and they make sure that the barcodes are exactly in the same position for every scan. And now we just place it there, push a button, we scan, we go. So that works perfectly. We now even have here a holder for the battery. So we can place the battery here. So it's one thing we can carry and then to the next plot.
Wow.
Sorry?
You even included an external power supply, I see here. Yeah,
yeah, we need to in the Alps. We
Absolutely.
really need it.
Yeah.
And that is working well. That is a, what is the company? Panasonic, I think. It's a Panasonic. Are we allowed to do advertisements? I don't know.
We are not advertising,
so
we
it's
are facts. Okay. No, but
so
it works.
An outdoor camping power station, and that lasts for, I think we can scan for 8 hours with one battery. So it's
also good that the power consumption of the whole system is considerably low.
Yeah.
Yeah, nice. And then yeah, so you had to make up your minds, you and the engineers, on how to make this system applicable for your demands and the environment you want to go into. So if we would now extend this question a little bit further, yeah, with all the knowledge you have gained now, if you were now to design a phenotyping system, yeah, may it be in the size of what you have now, so flexible and able to carry it around, or may it be much bigger? So just let's dream about, we don't have any fixed budget. Yeah, we can spend as much money as, or you can spend as much money as you want. What would be for your ecological trait-based studies, what would be the ideal plant phenotyping system, and what kind of major functionalities should it provide?
I think we need to talk about here the hardware and the software. For the hardware, I'm very happy right now with this machine. It's relatively light. I mean, when we go to the Alps, we already hired strong students to carry it. So it will be like 15 to 20 kilos, the whole thing. So that is quite a lot actually, but it's manageable because we can put it in separate parts. So that is manageable. What I would do is I would increase the scan area a little bit. So at the moment we can scan, I think it's 40 to 60 centimeters. And that would be nice to have this a little bit larger. Maybe, I'm not sure whether this is like technically possible, but maybe it would be nice to have a circular scan and to just have the scanner at a fixed point, and then somehow it is scanning a circle. I don't know. So that we can have this perspective, yeah, to have a larger scan area. That is something I would ask for if I had the chance. The other option would be
quick question, why would it need to be circular? Why couldn't it be?
I just thought about the handling. So we would just have something like a tripod where we have a device like that, and then there is the scanner, and it's just scanning a defined area.
All right.
I don't know. So it doesn't need to be circular. It was just an idea.
But
there is no specific reason why it should be circular. So that was just, maybe it would be nice to have this plant eye on a drone, which I think is too heavy. So that should be a large or a huge drone then, which I think is not available. But people use drones for similar things in natural systems. Yeah. But the resolution is lower, and I think the match between the 3D point cloud and the multispectral information is not as accurate in these other systems. So they don't really have a 3D information on these drones. They only have a multispectral for mostly information.
I mean, the advantages of having a proximal scanning system, which is quite close to the target, is always that you gain in resolution. So in absolute terms, in millimeter accuracy or sub-millimeter accuracy. However, current drone-based or UAV-based systems work certainly with a different measuring node or measuring mode, because the line scanning that the plant eye uses is not fit for being applied from large distances high above. You're scanning more or less a laser line that is invisible to the eye, but the further you get away from the target, the less likely, or the higher, the lower the signal to noise ratio, and at some distance you're simply losing sight of that laser line. So laser scanning is certainly not fit to be applied from drones, unless you have a very, very powerful laser. But well, you want to continue working with the plants also after the scanning. So that kind of high-powered laser would certainly.
So I understand that, but alternatively, maybe not. It doesn't need to be a drone, but maybe it should be a mobile device where we have this plant eye at the front, and then we have a constant speed of this device, and then we just go a gradient. That would be cool
that
too.
is very well possible. Yes.
So maybe we can think about that.
Very nice.
And then from the software side. So maybe then I specify my answer to the first part. I'd like to have this mobile thing with a constant speed.
Good. That's a straightforward answer.
The second, for the software, I think this hot control software is really great. It provides a lot of parameters that are useful that we can use right away from the machine, and we don't have to do a lot of calculations. We started to also work on the raw data, like on the three-dimensional so on the point clouds plus the multispectral information. And that is, we are at the very beginning to first we worked on voxels to get information like that to aggregate it into larger spatial parts. Now we are trying to use deep learning to extract more information and to relate these raw data to diversity of the plots and stuff like that. And that is clearly something that I'd like to improve and to be, in order to being able to separate plant individuals in a scan, to separate maybe plant species even in a scan. And that is something that I wouldn't expect from Phenospex in the end. That is something I would expect from us as scientists, that we work on better machine learning, artificial intelligence solutions that do the job. And as I said, we started, but we don't have results to show yet whether this can work or not. But I'm very optimistic, at least.
Well, you foreshadowed already a little bit of this topic in your talk when you showed the forest segmentation, or the random forest regression. There is also could be considered a machine learning algorithm.
But there we didn't use the raw data. We used the parameters we got from hot control.
Exactly, that was where one of the questions that had been sent in was directed at. I'm quickly reading it out. So in your study where you used the random forest regression, what were the input variables? All plant eye parameters or just digital biomass? How did you receive the relative importance of parameters for the prediction in the end?
So we used all 17. So there are 17 more or less parameters that we get from hot control that we considered as useful.
Physical and spectral.
Yeah.
Okay.
Yeah. So it's NDVI, biomass, leaf angle, all these things. So that are applicable from hot control, or that we get from hot control. And so that was the explanatory variables, and the response variables were like the origin of the communities and the treatment. And then you said that we could over time, or you saw that over time we were less able to predict the origin of the plots, but we were more able to predict our treatments. And that is something we hoped for, that this exactly is happening. So we used that and got these. It wasn't a regression, it was a classification problem. So that is, so we classified these plots to their groups with acceptable accuracy, I would say. And the variable importance is like how you do it in random forest. That is, you have these different measures for variable importance. I'm not sure which one we picked here, but that is the normal random forest variable importance that you get from the standard analysis in R.
So a dimensionless scale of.
So yeah, right. And we just ranked it. So we didn't work with the values itself. We just ranked it and found that mowing, of course, can be best predicted by physical features like plant biomass, plant height, and stuff like that. Whereas the fertilization is more these no morphological traits, and the reflectance traits, they were more predictive for the fertilization.
Oh, okay.
That's also interesting. Also in this strawberry project, I think the plant eye is or the digital phenotyping is very well suited to separate the to separate the effects of certain environmental conditions on certain traits. So we always found that wind and drought, we found different traits that responded to these treatments. And that is very relevant for our future projects, where we can really disentangle the effects, and then we scan a community and we can say, okay, here this force should have been active in order to produce this phenotype. And that is really cool. So let's see where we can go there.
It's also much favored by breeders, of course, selecting certain lines that respond in a certain way towards distinct environmental variables or stressors. For them, this information is also very important, and that goes, I think, in the same direction as you just explained. So in general, in the traditional plant phenotyping applications, G by E, so genotype times environment is a paradigm which is often in the center of those traditional plant phenotyping applications, for example, in plant breeding.
Yeah.
In your community studies, you investigate the G by E relationship on a community level, so to say. That means for studies focusing on community function and or traits, you would I would postulate that when trying to adapt G by E to ecological studies, G is not so much referring to the genetic information of an individual species or line, but rather more to the functional genetics represented by the different community members. So a pool, so to say, of genetic potential, which is, of course, also affected by the environment and maybe sometimes activated by the environment. Would you agree? And which other main components would you include as the center or target of digital plant phenotyping in the ecological context? Maybe management / disturbance, question mark? Sorry for these big questions. Maybe we separated. So first of all, would you agree that G by E yeah, the traditional paradigm here could be applied to ecological applications as well.
Mm.
Makes sense?
I think, yes, I would call it then C by E,
Mm-hmm.
community by environment. So we have different communities and how they respond in different environments. So I would translate it like that.
Mm-hmm.
Of course, as I said, we cannot separate plant species. And in the field, we definitely do not have any idea about the genotype. So
Mm-hmm.
that is just the limit of information that we have in the field. We can't genotype each individual plant individual there, not even each plant species. So first of all, we do not look at the genotype. We look at the phenotype, which is of course dependent on the genotype. But with this scanning technique or this digital phenotyping, we can't even look at individual species at the moment. So I hope, as I said, that may change. So I would agree that this C by E question that is central in ecology. And we need to learn and we want to learn how communities with different properties respond to the same or different environmental factors. And
okay.
So, yeah, I would agree with that point, that these questions are similar. And this leads to the other point regarding why do we want to know that? And here we can go back to the very beginning when we started to talk 45 minutes ago. We want to learn about general rules, how plant communities respond to perturbations, to climate change, to other threats of our biodiversity, of our natural systems. And I think the scanner can help a lot there, because now we can gather a lot of information within a short period of time. So in this glacier forefield that you see here, so there at the very back, there is the remaining of the glacier. And from there on, we can track the succession of community assembly. And so we did all the diversity inventories. We know which species grow where. We know which microbes grow where. We know about the soil properties, but we don't know about the biomass, for example. We don't know about the functional diversity. We don't know about reflectance properties, about the chlorophyll contents, which can be estimated by NDVI, for example. But these are very important properties of communities, and they are surrogates for very important ecosystem functions of plant communities. Mm-hmm. And I think by using the scanner, we are now a step further or closer to the real functions of plant communities. And that will then help first to learn basic ecological questions regarding succession, for example. And it will help us to learn about how communities respond to perturbations, climate warming, invasive species, pollution, whatever, in this functional perspective. And that is then a general finding, and we can predict the future fate of these communities under ongoing climate warming, under different scenarios. And that is, I think, the way forward. At least I hope so.
Sounds very good. Robert, at the end of our little interview, I always give the people a chance to outline conference attendances or future events or things to watch out from their working group. Is there currently a paper in the making already combining digital plant phenotyping and ecology? Is there a new project where you take part, or is there a conference where we can actually see you in the near future? I mean, conference-wise, currently it's a little bit difficult. However, many conferences work the way we do by offering video call or virtual conferences. So is there anything where we can see you in the future, or what is to be expected from your working group in the future?
Yeah, so regarding papers, we are working on the first paper where we just like introduce digital phenotyping into ecology. And here we will wrap up these common garden results more or less to show that manual measurements and digital phenotyping results correlate very well, so that this is a valid method and that we can track fast responses by plant communities using this technique. So that will, I don't know, so we just started, so it may take a little while until it's out. For conferences, I will apply for the GFÖ, Gesellschaft für Ökologie, which will be in Braunschweig this year. It will be online, but hosted by Braunschweig. And there, I think I will talk about digital phenotyping. Let's see. So, but that is my plan at the moment. And regarding projects, so now we have this wonderful new technique and these new options. And I'm writing proposals right now, and of course, everywhere the plant eye is included, because I think that is something we should do more and learn more about these traits of communities. And so it's ongoing.
Very good to hear. So also for you, Robert, if you want to stay informed on the world of plant phenotyping on an international context, you may want to know and visit our Facebook, LinkedIn, or Twitter page of the IPPN. There, almost daily news from the plant phenotyping world are being posted, and of course, we would be very happy if we would gain more followers. If you want to do that on a European level, please visit the same platforms, but on the emphasis, from the emphasis side, that's the European Plant Phenotyping Network. And also, I want to mention as the third organizer of this webinar series, the EPPN 2020 project that grants researchers access to plant phenotyping installations across Europe. And I want to highlight our next Phenomics webinars, which will be uploaded just after after we are ready today. It is on, and this is maybe also interesting for you, Robert, on semi-automated object detection workflows using UAV, so drone imagery. Yeah, certainly very interesting. This webinar will be provided by Samuel Oswald from Vito in Belgium, and we will in about 2 weeks have also a Q&A on the topics of this webinar. For now, I want to thank you again, Robert, very much for this interesting discussion we've had here. I hope you are not running late for the next meeting. And still, I want to cut the time here. We had 55 enjoyable minutes. Thank you very much.
Thank you very much. It was a pleasure to be here. Yeah.
It was a pleasure to host you. All the best for the future. Bye-bye.
Thank you. Goodbye. So I leave now, yeah?
Oh, you can leave and I just end the
okay, good. Thanks.
Okay, bye-bye to everyone else.
Nice talking to you.